So we're here, you all know that we're going to talk about next-gen sequencing, and in particular, we're going to talk about the challenges of DNA library prep, and we're going to present some solutions which are products that Lucigen has created to solve those major challenges. What I'd like to do is simply start with introduction to de novo sequencing, very, very brief introduction, but the idea of use that as a baseline to start talking about DNA fragment library preparation. Then we'll review Illumina DNA fragment library construction. We are focused on Illumina today because the kits that we've developed are built for Illumina sequencers. Then we'll talk about some of the challenges of DNA fragment library prep, and then we'll introduce a solution to that, which is our brand new NxSeq AmpFREE low DNA library kit. This really does solve many of the problems associated with fragment library preparation. And then we'll switch gears and we'll talk about another type of DNA libraries, which are called mate pair DNA libraries. And these are very, very different. So we'll go through an overview of mate pair and how they work and what they're used for. And then we'll talk about some of the challenges of mate pair library construction. And finally, we'll introduce the NxSeq mate pair kits. And again, they offer solutions to many of those problems. And of course, we'll end with a summary. Okay, I cannot see the full screen. Okay, there should be an option for you up under NxSeq Library Prep Webinar _ March 2016 PowerPoint. I believe if you click on that and click full screen on that arrow, it should open up to full screen for you. All right, so the first slide just talks about de novo next generation sequencing. Basically, this is doing whole genome sequencing on a new genome where you're trying to build and develop the sequence and put it all together. And basically, the process usually starts with DNA fragment library, okay? And what you end up with are many short read sequences that are assembled based on their overlap into contigs, okay? Now, the problem occurs when genomes have repeat regions in them. They often will not sequence or map very well. And so the contigs that you build are often not linked together. So here in this illustration, I've got 3 contigs that correspond to that region of the genome. But in fact, when you do the sequencing at the contig level, you won't know what order they're in. You won't know if they're next to each other, separated by a repeat. You really have no idea where they are in the genome. And so that's where mate pair libraries come in, and we'll talk about this in a lot more detail. But basically, mate pair libraries allow you to join contigs. Even though you might not get the sequence in the middle, it allows you to put them together, and you can order them and start to build scaffolds. So what people will generally do for de novo is they'll build mate pair libraries of increasing size, and that allows them to assemble more and more of the genome into fewer and fewer scaffolds, hopefully ultimately ending up with one scaffold. Now, that's an example of DNA fragment library preparation for de novo sequencing, whole genome sequencing. But there are many other applications for DNA fragment libraries. That includes resequencing, mutation SNP detection. Think of cancer researchers that want to identify mutations involved in cancer. ChPCR's another application, and then targeted capture / sequencing. So this is whole exome capture, targeted capture of your favorite oncogenes, whatever you want. Many of those kits include library prep in them. The kit we're going to talk about today can be substituted. You just have to watch out for how you're doing your targeted capture and your library prep. Now, the library step is absolutely critical, and that's why we're talking about it today. A poor library equals poor sequencing results. And this slide just gives you an illustration of how the Illumina library prep works. So you start out with genomic DNA, and usually this is large, okay? And it has to be fragmented. The Illumina instrument will not work with large pieces of DNA. It has to be made into small fragments before it can be sequenced. And so you can use multiple methods here. You could use sonication, or you can use enzymatic methods like fragmentase from NEB or shearase plus from Zymo. All these methods will allow you to chop the large DNA into smaller pieces appropriate for the Illumina sequencers. But those fragments that are produced are going to have ends that aren't blunt. They're going to need to be repaired. And so the first step in library prep is usually an end repair step, and then generally an A tailing. Okay, so you've got blunt end fragments with an A tail, an A overhang now. That's important because the next step is adapter ligation. And of course, Illumina's built the Y adapters. Okay, we don't need to go in a lot of detail there, but the Y adapters have an T overhang that's compatible with the A overhang of the DNA fragments. And so you'll use those to create some complementarity, and then ligase will ligate them together to produce this library ready for sequencing. Now the key here is that the fragment libraries, you want Y adapters at both ends. If you have Y adapters at one end, it's not going to work on the sequencing instrument. Okay, so it's very important that your library kit and your protocol be very efficient in making as many fragments with Y adapters at both ends as possible. Now, if you have limiting amounts of library material, you can do an amplification step, a PCR amplification step that uses primers for the red and green regions that will amplify these DNA fragments. We are going to talk about a kit today that doesn't require amplification, and we'll talk about some of the issues with amplification as well. And then once you've got your final library, you throw it on the Illumina sequencer. As simple as that, just throw it on. No, not really, but these libraries are compatible with multiple Illumina instruments, and I've illustrated the more popular ones here. And of course, we're talking all about library construction today. So there are some major challenges that customers and researchers have told us about when it comes to fragment DNA library construction. The first one has to do with the starting amount of DNA. Many kits require a large amount of starting DNA when you're not doing PCR, okay, when you're not doing an amplification step. So for instance, the Illumina PCR-free kit starts with a microgram of DNA. It's a lot of DNA, and many times folks don't have enough material to use that type of library prep without an amplification step. So one option is to introduce PCR, and a lot of kits use PCR to amplify those libraries at the end. Problem is bias, right? We all know that some fragments amplify better than others, and some just don't amplify really, really poorly. And so if you're looking at, for instance, a whole genome sequencing experiment, you've made a library where the fragments are supposed to represent the whole genome. You really want all those fragments to amplify equally, because if you don't, you'll have coverage across a genome that's uneven. You'll have regions of very, very poor coverage, some regions of very, very high coverage, whereas you really want it to be fairly even across the whole genome. It just means better sequencing analysis, easier sequencing analysis down the road. And then finally, inefficient library construction. This is another major challenge. As I said, the key is to get Y adapters added to as many fragments in that library as possible. What that means is you're going to have the most complex library possible, the more fragments, the more adapters you have attached to the fragments, okay? If you have inefficient library construction, that's going to decrease the amount of data that you can get from each sample just because fewer fragments are sequenceable. And then remember, this is, you might be able to sneak through with a poor library if it's the only sample you're putting on a run on an Illumina instrument through a flow cell. You'll probably get enough reads to make it work. But if you start to multiplex poor libraries that are barcoded, then you will compound the problems of analysis and sequencing depth and coverage. So really, it's even more important when you multiplex samples. Okay. Now, before I go on to this next slide, I want to get your opinions. So these are what we think is important, but I want to know what you think about, hold on one second, let me get this to work. Okay. What do you think are the most important features of a DNA library prep kit? Okay, so it's not really, it's basically what's most important to you. So you should be able to see all this. Everybody should be able to see the results. Please pick your favorite one that is most important to you. It'd be interesting to see what everybody says. It looks like low input amount is a winner so far. Keep going. Okay, high quality data is important, obviously. That's a good one. Fast protocol. Okay. Oop, oop. Looks like everybody's changing their minds. All right, well, this is pretty good. I'm going to stop here, finish it up, but it's cool because that's what this kit we're going to talk about does. It's really all about using low input amounts of DNA. All right, so let's get on to it. So those are the challenges, and we think we've built a kit for library prep that is um, really good, that is going to meet many of the challenges that we discussed. So the kit's called the NxSeq AmpFREE Low DNA Library Kit. And the bottom line is it requires only 75 nanograms of input of sheer DNA. You can go up to a microgram if you want, but really it takes only 75 nanograms of sheer DNA. And this, of course, is going to make many more samples accessible to you. You're going to have a chance to run many more samples with this kit. And as I said, it's based on excuse me, a no PCR protocol. So there's no chance of any amplification bias introduced if you're using 75 nanograms. If you want, we have a PCR step in the appendix. You can do PCR amplification of the library if necessary, but it's not necessary as low as 75 nanograms. Also, high efficiency reagents. We've made a library kit here that is super efficient at adding those Y adapters. Okay, so that means you're going to get more complex libraries and better sequencing coverage. Now, everybody's asking me how we did this. I mean, when you look at these library kits, they're really pretty basic molecular biology. But what we've done is we've optimized every step of the protocol to make it work as best we can. Okay, so we've optimized end repair, we've optimized A-tailing, and we've optimized the ligation reactions to really produce the most efficient libraries. The other thing we've done is it's a single tube protocol for all the enzymatic steps. So that's going to allow you to work with those smaller samples because there's no risk of loss if you're not transferring tubes or doing cleanup steps in the middle of a protocol. The only cleanup steps that we do, as you'll see, are at the end. Okay, so fewer cleanup steps mean higher yields as well. It's also fast and efficient, so it's only about a 2-hour protocol. That's going to get your samples onto the sequencer faster. And then it's also automation friendly. We haven't tested this directly, but we have built a multi-channel 96-well plate protocol that we've tested. We've included it in the appendix of the manual for the kit. And because of that, we know that it should be easily automated on a liquid handler. And again, the single tube reaction format will really help a great deal. These are the kits that we're selling. So we've got 2 library prep kits, a 12 reaction and a 48 reaction. And then we have 2 sets of NxSeq adapters that are compatible. These are the Y adapters are compatible with the Illumina instruments. And each one has 12 different adapters in it and a total of 4 library preps, enough for 4 library preps with each adapter. So 48 total reactions in each adapter box. So you need to, just as a reminder, if you are buying our kit, you do need to buy adapters or supply adapters separately. They're not included in the kit. We'll talk about costs and stuff at the end if anybody's interested. So here's a slide. Basically what I want to do now is just work through and give you supporting data for all those claims that I made at the beginning. So the first one is the FAST protocol. So here's our protocol. Here's the end repair and A-tailing, 40 minutes, reaction setup, another 30 minutes for the ligation, and then 2 AmpPure-B steps at the end. In total, it comes out to about 2 hours and 10 minutes with minimal hands-on time. Okay, Kappa's on the order of 3 hours. Illumina's on the order of 3 hours and 25 minutes. Again, they've got multiple cleanup steps sort of in the middle there. They do a size selection early and so forth. So that's going to lead to a lot of product loss or potential product loss. So it is super fast. Now I want to get into the data that actually supports everything I said. But what I want to do is tell you before we start that we've really looked at 4 different genomes in all our performance characteristics or performance analysis of the kit and the competitor kits. So we've looked at human, of course, because that's super popular and it's a big genome. And then we've looked at staph, rotobacter, and E. coli. We picked these organisms because they have very different GC contents. So staph is AT rich, it's only got 24% GC. Roto is 68% GC, so super GC rich. And then E. coli kind of splits the difference right in the middle. The other thing I'll let you know is that all this data you're going to see is done on the MySeq. So the MySeq provides plenty of sequencing reads to cover these bacterial genomes very well. It's not going to give us enough depth, enough sequencing reads to cover the human genome very well, but it's still a very good indicator of how the quality of the libraries. And so you'll see that data as we go through. So the first claim is that the kit requires only 75 nanograms of sheer DNA input. So basically this table just shows you how much we're getting from libraries from humans, staph, and Rhodobacter when we started with 75 nanograms of sheer DNA. So these are the total yields in nanograms in a 20 microliter final library, okay, final volume. You can see there's a lot of DNA there. The lowest was 23 nanograms. And then when you figure it out to concentrations, these are running between 2.83 nanomolar and 4.3 nanomolar. Now the important thing is that when you're running a MySeq, you need to add 5 microliters of a sample or a multiplex sample where the total DNA concentration is 2 nanomolar. The library is at 2 nanomolar. And so you can see that all of these libraries are more concentrated than needed, and so they all have to be diluted to get to create a 2 nanomolar stock for loading. And what that equates to is it means that there's enough of each library to actually run 5 independent runs on the MySeq without multiplexing. So there's plenty of material here from 75 nanograms of DNA. Now on to the efficiency measures. Okay, I made a big deal out of that library efficiency is important. And so what I wanted to do first was just tell you how we measure library efficiency. And again, the data that I'm going to show you, everybody was done, all the tests were done the same day, the same way. Okay, so it's apples to apples comparisons. Some people might do it a little bit differently, but this is how we do it. So basically the question is, what percentage of the fragment library has the adapters correctly ligated to both ends of the fragments? Okay, you can have unligated, you can have fragments with adapters at one end. These, of course, are dead-end products. They will not work on the Illumina sequencer. Okay, so the more you have of this, the better. So we use basically a combination of qPCR and fluorescence quantitation to come up with an efficiency measure. And in fact, we use a kappa kit for doing this, which you can buy as well. And this is a little bit overly simplified approach, but I wanted to just make it simple so that it's easy to get through. But basically, you can either make or buy an amplified library, okay? So either the standards that come with the kit are amplified, or you can take your library and amplify it. And then you quantitate it to get a fluorescent measure of how much DNA is in that library. Because the take-home is, after you amplify, it's going to be a 100% efficient library. I just did air quotes here. Basically, all the DNA, the only stuff that will amplify has the adapters at both ends. So that's your control. That's 100% efficiency. And then you quantitate how much is there by fluorescence. Then you can either serial dilute that amplified library and use it as standards, or you can use the kit standards. Okay? Then you go forward and do a qPCR with primers for P5 and P7 regions, okay, to amplify by qPCR. And of course, you'll do your unknown as well. And then you compare your unknown to the standards to figure out how much DNA you have there by qPCR. And again, it's not all the DNA, it's just the DNA with the adapters correctly ligated. So in this, and then you compare, so in this example, for instance, the qPCR said we had 1.5 nanograms per microliter. I'm making these numbers up, okay? It's just an example. 1.5 nanograms per microliter, and by fluorescence, we had 10 nanograms per microliter. And so you divide those two, and you come up with a library efficiency of 15%, okay? So that's basically how we do it. It's a little bit more complicated than that, as I said, but that's basically how we do it. So when we did that with libraries prepped with the NxSeq AmpFREE kit, the one we're talking about today, our kit, in red, the Kappa Hyper kit, which is in dark gray, and the Illumina TrueSeq PCR-free kit in light gray, these are the numbers that we saw, okay? And we've done this multiple times, and we see this, basically all these trends repeating over and over again. But these are the percent of correctly ligated fragments in the library. And so when you actually add in the numbers, you can see that sometimes we're double kappa, much, much better than kappa, but really a lot better than the Illumina instrument. Remember, we're loading here 2 nanomolars or a combination of samples that equal 2 nanomolar, but they're all added equally in molar amounts to the sequencing run. And so, you know, you look at this and you go, holy mackerel, why is Illumina so low? Well, in fact, everybody knows this. This is not unheard of. And the reason is that, well, I don't know what the reason is, but the bottom line is what researchers will do before they do their sequencing run is instead of loading a 2 nanomolar stock based on fluorescence, they will do qPCR on their Illumina libraries. And the reason they do that is not to amplify them, but to actually quantitate how much of the DNA fragments in there have the adapters ligated correctly. And then they'll basically overload the instrument with more DNA than 2 nanomolar, 5 microliters at 2 nanomolar. They'll overload it so they can put more fragments on there that can bind to the flow cell and be sequenced. So it adds a step. It works, absolutely works, but it adds a step and it adds cost and time. So with our libraries, they're so efficient that you really don't need to do that qPCR step. Um, Once you get comfortable with them, you'll see that that's true. So what does it really mean? Efficiency is great. That's a great bar graph, right? Looks like we're very good. But what does it really mean when you get down to sequencing? And so what it really means is you get more sequencing reads from each library prepped with the NxSeq AmpFREE kit. Okay, and this slide just grabs the total reads. Here's staph data. Here's E. coli data. Here's, again, this is a multiplex run, okay, on a MySeq. Here's the AmpFREE kit. We got 5.7 million. Here's E. coli, 4.3 million in this particular experiment. Kappa hyperprep, fewer, many fewer with E. coli, and the Illumina TrueSeq, very, very few reads, okay? A little bit better with the E. coli. And in red, I've put the decrease compared to, percent decrease compared to the NxSeq AmpFREE kit. So you can see there's a huge difference. And again, you might be able to get with a bacterial genome, you might be able to get enough reads, but as soon as you start working with compromised samples or you start working with, start multiplexing more samples, these limited numbers of reads are going to kill you. They're going to really impact the data quality and your data analysis. So this is all clean DNA. Let's look at a sample that's a little bit more challenging. So people are always asking, does it work with FFPE? Well, in fact, it does, and it works really well. So here's an example of library prep and sequencing from FFPE samples and matched normal genomic DNA. So what we did was we bought normal genomic DNA from BioChain, and they had a matched FFPE human kidney tissue. Okay, so both kidney tissue DNA, or excuse me, this is kidney tissue DNA, but it's normal, and then this has been fixed. Okay, and it is obviously FFPE fixed, so it's formalin fixed, paraffin embedded. Terrible stuff to work with. If anybody's worked with it in the audience, you know how terrible it is. DNA, when you extract it, comes out fragmented, cross-linked. You really have to do a good job of getting the cross-links removed and so forth for it to work in any type of assay, much less next-gen sequencing. So what we did was we had the clean DNA as a control, and then we took the FFPE tissues and we extracted the DNA from them using the QIAGEN All-Prep DNA RNA FFPE kit. And then we took both samples and we sheared them down to about 250 base pairs and made libraries using either the NxSeq AmpFREE kit or the Kappa hyperprep kit. And again, we used our recommended amounts for normal DNA, which is 75 nanograms for the low input. But we were a little bit worried about FFPE being super high, super poor quality. And so we also bumped it up to 150 nanograms to see if that made a difference. Then with the Kappa kit, we did the normal DNA at 250 nanograms. That's their low recommendation with no PCR. And then we did 250 nanograms for the FFPE DNA as well. That's what they recommend for FFPE DNA as a starting point. And I should also point out that, if anybody's working with FFPE, you know how precious that DNA is. So if you can get away with using less DNA in your library prep, that's going to make your FFPE samples, your FFPE extracted DNA, go much, much farther, okay? So that's a valuable point, okay? It's an important point. So let's look. Here's total reads in the second to the last column, and here's mapped reads in the last column. Total reads, 2.1 million for clean genomic DNA with the AmpFREE kit, 1.7, almost 1.8, 1.7 with the 150 nanograms, okay? Nice, a slight reduction in what we'd, compared to the control, but really very, very good. And then the same pattern held true with the mapped reads, okay? And these are repeat masked, so these are only the reads that will match to the non-repeats, okay? So the numbers are running here. Then the Kappa hyperprep kit, 250 nanograms, 1.5 million. This is a repeat of what we've seen before in the data I just showed you, so fewer reads. And then with the FFPE, they had a reduction again, down to 1.2 million, so about 500,000 fewer reads from the FFPE genomic DNA compared to ours. And again, that's using almost four times more starting material. So bottom line is the kit works very well on FFPE DNA, just like it does on clean genomic DNA. But it's, again, and they're valuable samples, so using less is super powerful. So a couple questions comes up. Why was bumping up the input DNA not making any difference? Could it be the amount of adapters to workflow not sufficient? No, in fact, it's not. Basically, remember what's happening is we are loading on a 5 microliters or some fraction of that, of a 2 nanomolar stock onto the MiSeq. So everything is normalized to 2 nanomolar. So if the library efficiency is the same between the 75 and the 150, then basically once you've normalized to 2 nanomolar, you're adding the same amount of sequenceable fragments and therefore the same amount of sequenceable reads. So in fact, it's really kind of a good control that things are very consistent. Okay, hope that answers your question. As I said, we can go up to a microgram if you want to. All right, and then just a few more examples looking at the human genome. Again, very large genome. This is, again, a multiplexed sequencing run, so we can't get full coverage here. You'll see the genome is covered, fraction coverage is about 0.1x, and then average coverage across the genome is 0.15x. So exactly what we would expect, but the mapped reads are very high. This is, I got to remember this, this is unmasked, so this is mapping all the reads to everything. And you can see that it's 95% mapped reads, very, very good. The read length is what we would expect since we did 2 by 150 chemistry. And then this is just total bases, and as we said, the coverage is what we'd expect for a MiSeq run on a human genome library. Then looking at staph and rhodobacter, okay, much less complex genomes. They're only 2.8 and 4.6 megabases, so they're much, much smaller, okay. And again, 2 different GC richnesses, if that's a word, of the genomes, okay, and 1.2 million, 1.3 million reads here, 3.9 million reads here. Again, very good mappability, greater than 92% for both samples. Read lengths on par with what we'd expect, and then coverage is much higher now. So we've covered basically the whole genomes of both bacteria. We've got 62x coverage with staph because we've got fewer reads, and then 117x coverage on average with rhodobacter. So obviously, the MiSeq is built for lower complexity, higher, easier genomes and more targeted sequencing. So this is a great example of how well it can work on a MiSeq and get full genome coverage of a smaller genome. Do we have any data for lower input than 75 nanograms? We do. We've gone down as low as 50 nanograms without any PCR amplification. And we're pretty happy with those results. But it's not consistent enough for us to be super happy. So we've put our lower limit at 75 nanograms. Now, as I said, it's not required with this kit if you stay above 75 nanograms, but you can always add a PCR amplification step at the end. So that's in the appendix of the protocol. You can check it out. We recommend a master mix from another company to do that, and it works great. You can go very, very low. I think we've gone down as low as 10 picograms and feel good about it. But again, we're going to be launching a new kit pretty soon that will include a PCR step as well. So is 75 nanograms input or sheared to the target size? That is 75 nanograms of sheared DNA that goes into the library prep. Okay, so that's the starting material after shearing. Okay, already down to the right size. Yeah, so it's very hard to, everybody has different instruments for shearing, and they all, do it differently. So some might require a little bit more DNA, some might require less, so on and so forth. So we basically, so that we could be very clear and compare everything very clearly, we stuck with the amount of sheared DNA. It's also easier to quantitate, and the measures are usually more accurate. So that's the reason why we talk about it in terms of sheared DNA. Okay, so then the other thing is you don't want any bias in your sequencing reactions, okay, in your sequencing results. And so what we've shown here are staph, E. coli, and rhodobacter, and I've just put them on, and it's basically normalized coverage on the y-axis and GC richness for the genome on the x-axis. And the bottom line, what we're presenting here, the normalized coverage just means it's the average coverage of all windows with a particular GC content. So right here it would be all the windows at 20% GC compared to the, and so it's the coverage there divided by the overall average coverage of the whole genome. And so what, if in a perfect world, this line would be straight across at 1 in all these samples, okay? And of course, we've compared ours to Illumina and to Kappa Hyper again, as you can see. In this case, Illumina's dark gray, okay? So what you can see is that everybody kind of performs the same way. Rhodobacter is pretty demanding. It's got some super GC-rich regions, and so everybody kind of drops off in the same way. So I'm not making any claims here that we're better or worse, but we're all pretty consistent in GC bias or potential for GC bias. And then if you look out here, you look at these wavy gravy stuff, and over here as well, it looks terrible, right? Well, that's just because there's so few regions of the genome at that, in this case, low GC or high GC content, that the number of reads, there's just a lot of scatter in the data analysis and the normalized coverage. So don't worry too much about that. All right, so then I want to switch gears now. So any questions now you want to get out of the way or save till the end, or you can also save them to the end as well. Okay, nobody's typing, so I'm going to keep going. I want to switch gears now and talk. We talked a lot about DNA fragment library prep. I want to talk about mate paired libraries now. As I said, mate pairs are a way to connect contigs. If you're doing de novo sequencing or whole genome sequencing, it's a way to connect contigs to start to build scaffolds. Okay? And it's really pretty cool. It's a lot more complicated than making a standard DNA fragment library, but it's a pretty cool technology. And so I'm not sure if everybody is familiar with it, so I want to run through basically how it works. And keep in mind that this is a oversimplified version, but it gives you the take-home message. Okay, so here's your favorite genome. It's a stretch of DNA, dark gray on one side, light gray on the other side. And if you did a DNA fragment library on this example, you might get sequences at the left end and you might get sequences at the right end of the genome. So these would be your two contigs that you could build from this stretch of the genome. This middle part is not sequenceable or it's not mappable very well because it's maybe a high repeat region. There's a ton of repeats there. So remember, these contigs, you've got these contigs, light gray and dark gray contigs, but when you do this sequencing, you don't know if they're connected to each other. They are because of the drawing I made, but we don't know that when you actually look at the raw sequencing data. So the goal is to see which contigs belong next to each other. And so that's where mate pair sequencing comes in. Basically what you try to do is you start by isolating large fragments, so 2 to 40 kb pieces of the genome. And you use different sizes depending on the size of the repeats in your particular genome. And then often people will do mate pairs of, say, 3, 5, or 3, 8, and 20 kb, for example, to try to assemble their genome. But you can do whatever you want, and we can provide advice on that and so forth. But the bottom line is the goal is to have fragments in this library that are larger than the repeats you need to span, so that the fragments, as you can see in this illustration, could span that repeat or non-sequenceable region. Then what you'll do, and again, an oversimplification, is you add sequencing primers to the ends. Then you digest away the guts of that fragment. You're left with short sequences at both ends of the molecule. And then you will bring them back together. You'll ligate them together. And in our kit, we add in a junction code sequence so that you know what's left and what's right of the junction code. Then, of course, you'll PCR amplify this region and throw it on the Illumina sequencer. You'll get sequence in light gray here, and you'll get sequences in dark gray here. These correspond to this end and that end. Then you go back and you do your mapping, your bioinformatics, and you'll see, oh, okay, so the light gray sequence mapped to this contig, the dark gray sequence mapped to this dark gray contig. I know that these two, because I know that this mate pair came from the same fragment, I know that these two contigs must be adjacent to each other. So that allows you to build and start to build this scaffold. Okay? You won't get the sequence in the middle, but you will know that these two contigs are next to each other. Pretty cool. Now that's an example of using mate pair for de novo genome sequencing, where you're trying to build contigs into scaffolds and to assemble the whole genome. There's a lot more you can do with mate pair as well, and this slide just illustrates that. So one, you could do virus or transgene insertion sites. You could ask, where is a virus in this particular sample? Where did this, I'm making a transgenic plant. Where did this gene go in my transgenic plant? I need to know that. And so you can use mate pairs to do exactly that. So in this example, here's a mate pair fragment. there's a piece in the virus or the gene, and then there's a piece in the adjacent chromosome. Same way in this direction as well. And so you can use mate pairs in this case to map these guys. Say, hey, oh, this one is in chromosome 5 of this particular sample. You can also use it to analyze structural variation. Same concept. Here's chromosome 1 translocated and joined to chromosome 2. Mate pairs can span that junction, and you can do those mate pairs, and if you have a good quality library, you'll believe this. You'll know that chromosome 1 is now joined to chromosome 2. Now you might say, well, wait a minute. If I just do fragment DNA libraries, I can do the same thing. I can sequence over this junction, or this junction, or this junction, and I can say, hey, I know that this is what's happening. Well, the problem with that, or the disadvantage of that, I should say, is that it takes really deep sequencing to have any confidence in your junctions, okay? So mate pairs, because they're larger DNA fragments, seriously reduce the complexity of the sequencing you need to do. So it makes it much simpler. You don't need to go nearly as deep to get that information. And you'll have, and in fact, remember, you'll have you should have a lot of mate pairs that actually span that junction, whatever it is that you're looking at, because they're going to be starting at all different points along chromosome 5 and ending at all different points within the virus gene. So you'll have many examples that will, again, boost your confidence that that virus or gene is inserted there or this translocation occurred. All right, so what are some of the challenges of mate pair sequencing? So, excuse me, the big one is chimeras, okay? Chimeras are basically when you add your sequencing adapters, your different fragments, and what you get is something that has the two fragments joined together with the adapters at both ends. This is obviously a false mate pair or it's a chimera, okay? So when you sequence this end and you sequence this end, you're going to think that these two fragments, these represented one fragment, but in fact they don't because they're from different parts of the genome, okay? So that's bad. Don't want to do that. This is the correct situation where the sequencing adapters are added to both ends of each fragment, okay? That's obviously problematic. Now the other problem is repeat size. So as I said, the mate pairs are designed to span repeats so that you can attach contigs to one another, link them together. So in this particular case, it's the dark gray regions being linked across these repeats. And PacBio, of course, claims they can get across all these things, and they will get across many of them. But in this particular example, there are many repeats that are very large. PacBio can do maybe 20 kb long reads. They won't make it across this repeat, so they won't be able to join this contig with this contig. Okay. Same thing. Here's a 12 kb repeat. Illumina makes a mate pair library, goes up to about 10 kb. They'll never span this repeat size, and this big one is, of course, out of the question. Okay. But we're going to introduce to you the Lucigen NxSeq 20 kb or the long mate pair library kits, and the NxSeq 30 to 40 kb mate pair library kit. Okay, and these have the ability to make mate pair libraries from 2 to 20 kb in this case, and 30 to 40 kb in this case. And those obviously allow us to span many more repeats independent of their size. We can get across much, much bigger repeats. So here's basically how the NxSeq long mate pair library kit works. And really what it is it's optimized to try to produce the correct mate pairs, okay? So basically it starts by shearing the DNA in the large fragments. Then you'll ligate on adapters onto the ends that are then complementary or compatible with our coupler that we've designed, okay? And so you ligate and you basically make a circle, or it's no basically, you make a circle, okay? And then what you need to do is to use enzymes, restriction enzymes, to remove the internal pieces of that fragment, much like I showed you in that simplified illustration. You remove the major guts, and then you recircularize and you add that junction code. And this is basically the molecule that you're left with. Then and we'll go into that in more detail in a second. Then you use PCR to amplify from here and here, and you make these fragments. They're ready to go. You put them on the sequencer and get your mate pair data. Now, I've talked a lot about chimeras being important, okay? And I want to ask you how important detection of chimeras is, okay? Here's another poll. I'd like you to vote again if you can. Basically, how important is detection/identification of chimeras in your mate pair libraries? Now, if you haven't done it, of course, you don't really have an answer, but I'd love to hear what you think because um, we think it's really important and we want to make sure you agree, bottom line. Okay, ooh, 3, 2, okay. All right, okay, cool. I'm going to stop there. It looks like um, it's very important to 60% of you, important to 20% of you, and then another 20% don't know. Excuse me. So, cool. Thank you for that information, and that's awesome because, as I said, that's what this kit is optimized for. Okay, it's optimized to um, produce real, true mates. So here's how we do it. Basically, all those steps that I told you in the library preparation that I simplified and presented in that previous slide, they're all there to make sure that what comes together, what you see are sequences from ends of the same fragments, okay? And so what we've got in that final amplified product, you'll have a chimera code. These are unique sequences in the couplers, and we have 12 couplers in the current kit that allow you to differentiate between weird chimeras that occurred during the coupler ligation. Basically, if this is a sequence 1 and this is a sequence 2 barcode, you might have another that's a 3 and a 4. So if you had two couplers with two different fragments ligated in between them, so you've got a 4 fragment or 4 molecule circle made, those would obviously give you false mates. And we will see that when we do the final sequencing because the chimera codes will be incorrect. It'll be, this should be in a true mate here, 1 and 2 sequences that we detect. In that case, it might be a 3 and a 4 from a different coupler. So you know that something went haywire there, that you had a bad mate. Okay, so with this system, we then throw it on the sequencer and we get a left read that hopefully goes through the junction code. You get a right read that hopefully goes through the junction code. So that allows you to tell which side is the left mate and which side is the right mate, okay, in this illustration. Now, this kit, as I said, is optimized, so the mate pair efficiency is greater than 95%, okay? And we can detect and eliminate many of the false mates with these chimera codes. And of course, I told you about the junction codes. And with this long mate pair library kit, we can do libraries up to 20 kb with no cloning involved. We have a 2 to 8 kb protocol that uses AmpFREE beads for size selection, and we have a 20 kb protocol or 10 to 20 kb protocol that uses gel purification, okay? So you can pick the size that matters to you, but none of them require cloning. And of course, we've got multiplexing capabilities as well built in. Okay. Question here, do you make target specific or custom library prep kit? I'm going to assume that's the Fragment DNA library prep kit, and the answer is no. We don't currently make any custom kits. If you need special formulations, such as you want everything, you want a much larger kit and you want it in bottles instead of tubes, we can definitely do that if you need that kind of volume. We don't make any kits for targeted sequencing or Amplification right now, but if you need them, I'm going to give you my email at the end. Shoot me an email, tell me what you're interested in. I'd love to hear. So I hope that answers your question. Okay, now this slide just gives you an illustration of the mate pairs that we detected in a couple libraries. So here's an 8 kb AmpFREE bead selected library, and here's a 20 kb size selected by gel library. And basically, these are the number of mate pairs that we detected. Here, around 8 kb is the peak. Here's around 20 kb is the peak. And then these are just the distance. So you want everything to sort of focus around the size selection that you did. The more you have there, the better off your library is going to be. And this is very standard peak distribution that you would see when you do this type of experiment. So all's looking good here. I really want to show you some data as to how this works and what kind of difference it makes for you. So I'll show you that in a second. Excuse me. So the last kit I want to tell you about is that cloning kit. So this is the NxSeq 40 kb mate pair cloning kit. This is a much longer protocol. It's much more involved, but it's built to clone up to 40 kb inserts using now a phosmid backbone. So it's all the same, right? You take a 40 kb fragment, you clone it in, you digest away the guts, you rejoin, you do the PCR amplification, you do the sequencing, just like I showed you a a second ago. But the difference here is that you actually have to make clones. And so increases the time of the kit or the time of the protocol, but what it does is it gives you a backup. So every clone now you can archive, if you want, to save each of these phosmid with their 40 kb inserts. So if you get some neat data or you want to look at one of these particular fragments in particular or in detail, you can do that. You can go back and you can sequence that one really deep and figure out exactly what's in that 40 kb insert. So it takes a little longer, but it gives you that archival capabilities. So now, just to give you a comparison between the NxSeq MapePair technology and the NxThera MapePair technology, basically we can do up to 40 kb depending on the kit that you use. The Illumina NxThera is built to do 2 to 10 kb, so as long as you have small repeats, that's fine. But if you have any larger repeats, you're going to need larger MapePair capabilities. Our MapePair efficiency is greater than 95% true mates, okay? With the NxThera, it's about 5 to 10%. So the ability to detect and to prevent false mates is obviously very important. You all said that in the poll that I just did. So this number is very, very high, and it's really valuable when it comes to doing the analysis. Finally, Chimeras. Okay, these are true mates. Ours are largely prevented, so the way that we've set the library protocol up, they're largely prevented. And again, because of the way we've built the couplers and the junction codes, we can detect them as well, most of them as well. And then with the NxThera, there are many, many of them, and there's no way to detect what's a false mate versus a true mate. So then finally, this last slide just gives you an illustration of microbial genomes that were tried to be finished. They were attempted to be closed and finished using the NxThera MapePair technology and all the data was published in this journal article, and so it's down here for you to look at. So what we did was we said, okay, well, they got 0 out of 9 with NxThera. They weren't able to close any of them. So we said, well, what can we do with our technologies? So we went in and we looked at 7 of those 9 genomes, and we, in fact, were able to close all 7 of them. So I wanted to walk through that data really quickly as here at the end. So here are the 7 genomes that we looked at, whole variety of different sizes, different percent GC content, number of repeats and types, size of the repeats. Some are as large as almost 38 kb in strep, really big repeats. And then these are the number of fragment library contigs that were built off of fragment DNA sequencing for these genomes. So they range from just a few contigs, which is pretty good, from 13 all the way up to 123 in this case. The other thing I want to point out is researchers have tried to use PacBio to close the Staph aureus genome, and they weren't able to do it with the PacBio. Okay, so again, some of those repeats are just so large that you can't get through them even with PacBio with their long-read technology. So that's where the MapePair will come in. So there's a question here, just curious about what the duplication rate looks like with the AmpFREE kit. Okay, we'll come back to that later. I'm actually going to need, so the duplication rate should be very, 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 very low. I'll just answer it right now. Should be very, very low. I don't know the answer to that. One of my colleagues might be able to pipe in and give you a real answer to that. But the bottom line is, since there's no PCR, the only amplification you should hopefully see is from one fragment being made single-stranded, okay, and put onto the flow cell. So when that happens, both fragments have the capability to hybridize to the flow cell, and then they will both be bridge amplified and produce a cluster if all goes well. So I would say 2 copies is the best you could do. And again, they're going to be slightly different fragments, though, because the orientation of the, okay, that makes sense. Good. I'll stop talking. Good. Sometimes I heard that some of the fragments can actually, after the bridge amplification, can kind of come off and go seed another cluster, which would be a bad thing, but that's independent of us. Okay, so here are the genomes that we looked at, and again, they weren't closable with the NxThera. So we took and did a fragment DNA library with NxSeq and our long MapePair library technologies. We used spatial bioinformatics to help close all the data, to assemble all the data that we got. And it worked beautifully. Here's a reference that you can look at, okay? And the bottom line is we were able to close all 7. So here's what the data looks like. Again, here's the number of contigs that we started with. These are the scaffolds after using, including the mate pairs, okay? and using spades to assemble. So we had one scaffold in a couple cases, so we pretty much put it all together. A couple with just two, and some that looked a little bit more complicated, 4 chromosomes and 5 plasmids for thermos. Then we added in manual curation. We got down to 1 for everybody but thermos. Okay, and then final finishing requirements were a little bit of PCR in a couple cases and a little bit of Sanger sequencing. Otherwise, the manual curation was good enough. And with thermos, we ended up with 1 chromosome and 4 plasmid. And this is pretty cool. So obviously, I'm not going to show you a lot of sequencing reads or anything like that, but this is what it all meant in terms of building and assembling a genome. It worked. And just to show you, this is a fun slide. We're not going to go into any detail here, but this is the Thermus aquaticus genome that was finished. So here's 1 really big chromosome, and then here's the 4 megaplasmids. Now, I expect there'll be a quiz later about what all these different things are inside the genome. I'm just kidding. But this is what Thermus looked like when we're all was said and done. Pretty complicated, actually. So I want to just get to the end here and just summarize now. So we talked a lot about the NxSeq AmpFREE library kit. Basically, it is a PCR-free kit that can use as little as 75 nanograms of input DNA without any need for that PCR step. So if you're worried about any potential bias in your samples due to PCR, you can avoid it and use as little as 75 nanograms DNA. And again, what that means is that's going to allow you to get more sequencing data out of every sample. It's going to allow you to multiplex better because you've got higher quality libraries. All of that just compounds to give you more and more sequencing data and better and easier analysis. Also, we talked about the protocol. It's fast, right? We think it can be easily automated. That's going to save you time. And the price. So the price, I've priced this very, very aggressively, and that's going to help you spread your budgets out. And we can talk about that if you're interested. The mate pair kits, we talked a fair bit about that at the end here. Basically, the whole take-home there is, the key for mate pairs is knowing that those short reads that you get come from the same large fragment, okay? If they don't, your analysis is going to be messed up. You're not going to get the quality of the analysis that you need. You're going to have a lot of false mates, and you're going to be trying to put things together that don't belong. And so with these kits, we've got greater than 95% true mate efficiency. And remember, we can detect the false mates with the chimera codes and the junction codes. And so we know what's real and what's not. And so that makes your assembly super efficient and much more accurate. And then finally, just as a take-home, remember, we talked a lot about de novo genome sequencing with and using mate pairs to close the genomes. But mate pairs can be used for a lot of different applications. Wherever you want to try to map something like a transgene or a viral insertion or structural variation in a genome, it's all good for that. And again, the reason you want to do that with a mate pair instead of just fragment libraries is that you don't have to sequence nearly as deep. And you'll get multiple fragments, mate pairs, that will span those junctions, whatever you're looking at. There'll be multiple versions of that junction that you'll see that'll, again, boost your confidence as to what you're seeing as being real. Okay? So I'm going to stop here, and we've got time for questions. Okay, we've got about 5 minutes, and I see there's a couple questions there. I just want to point out, here's the Lucigen Tech Support email and phone number. We've got awesome tech support. I highly recommend you contact them with any questions about this product or any product you might see. If you're interested in more about this kit, that maybe pricing or whatever, you can contact me. I'd love to hear from you. If you have ideas for other kits, love to hear that as well, or any other particular issues that you're facing that are challenging to you. I'd love to hear about it. And here's my email. So, okay, what's the insert size of the final constructed sequencing libraries produced by your mate pair kits? That is a great question. I mean, they're all great questions, but that's a great question. So we use a partial restriction digest mix of enzymes basically to chop out all the internal fragments, all the internal sequence there to generate those short ends that are ligated back together. It's extremely variable. It depends on the genome. I really don't know what the overall size is, because if they get too big, we don't span that junction code. We tend to throw them away after the sequencing results. Some of those are real, but we tend to throw those away after sequencing because we didn't cross the junction code because those two fragments were too big. So it's all dependent on your genome. We actually are developing a new mate pair kit right now that is going to make that step actually a lot easier. We've got a mix of enzymes built in that you don't have to do any optimization for your particular genome. You just use it and go, and that'll make life a lot easier. And we're doing some other things to shorten up the protocol as well. So look for that to come out short time, hopefully, probably probably September or so. Yeah, summer. Look for it in the summer. But we're working on that hard, and we hope to have that out. Well, the other thing I should mention about the MatePair kits that we currently have, basically they work best on a MySeq. And if you want, you can contact our tech support to find out why. But we wouldn't recommend using our current kits for HiSeq, real deep coverage on the HiSeq. And it has to do with colors and cluster identification and so forth. But I'll just leave it at that. So I hope that covers everything. Anybody have any other questions? Oh, do you want to know about pricing? I'm going to tell you about pricing. So as I said, I've been really aggressive with this kit. Now, this is pricing in the U.S. through us directly. Pricing from your distributors may vary, blah, blah, blah. Okay, but if you buy from Lucigen, the pricing is set. So for a 12-reaction kit, it's $240 at list. For a 48-reaction kit, it's $921, okay? And that basically equates to $20 a library prep or $19 a library prep. Much, much less expensive than the other kits that we've talked about, okay? The adapters are sold separately. You need to buy those, or you can use any TrueSeq adapters that you have. But our adapters are actually juiced a little bit, so if you really want the best performance, I'd recommend you use our adapters when you can. But they're priced about $3 a library prep. So all in all, much less expensive than the competition. Final thing I want to mention is that we have a promotion going right now for the small kit. You can buy the small kit at 50% off, so that's $120 for 12 reactions, or $10 a library prep, okay? So, and that's going to end at the end of April. So if you're interested, I highly recommend you pick up a kit and give that a try, okay? Yeah, the end of April in 2016, yes. My colleagues telling me it's 2016. I think we all know that. I'm not going to go through next year. So recommend you, if you're interested, give it a call. If you need larger samples or something like that, shoot me an email. I'm happy to talk to you. All right. Oh, the other question is, are the slides available? If you send me an email, I can send you a PDF of the slides right now. The other thing is we are recording this webinar, and that'll be posted to our website as well. So you can grab the recording and look at the slides that way as well. So thanks again for your time. Really appreciate y'all coming out and listening. I know you're busy. And as I said, if you have any questions, let us know. Thanks, and have a good rest of your day.